Welcome back to Web Conspiracy, the show where we take those late-night, doom-scroll headlines and ask a very simple question. What's the actual story here? You know the vibe. You see a clip, it sounds alarming, the comments are on fire, and suddenly it feels like everyone's either panicking or dunking on everyone else. Today's claim is a big one, and it's been bouncing around online for a while, that a COVID vaccine used HIV and somehow infected people with HIV. If you've seen a short video snippet about an Australian vaccine and thought, wait, what did I just hear? Stick with us.
Yeah, because the way that claim is usually phrased makes it sound like someone literally put HIV into a vaccine and then people caught HIV from it. And that's not what happened. But I also get why the wording freaks people out.
Exactly. So we're going to slow it down and walk through the timeline, what the scientists were trying to do, what they actually used, what went wrong, and why the project was stopped. And spoiler, it's less of a scandal and more of a very normal, very important example of clinical trials doing their job.
Also, it's a perfect case study in how the internet can take one true sentence, chop off the context, and turn it into a completely different story.
All right, let's time travel back to early 2020. Remember that stretch when the world basically felt like it hit a pause button? Empty streets, hospitals under extreme pressure, everyone refreshing the news, and researchers all over the planet sprinting to find vaccines and treatments for this new coronavirus, SARS-CoV-2.
And there wasn't just one vaccine team. There were lots of approaches happening at once: mRNA, viral vectors, protein-based strategies, basically different ways to teach the immune system what to look for.
One of those efforts was at the University of Queensland in Australia. The team, led by Professor Keith Chappell and colleagues, was trying a technique with a memorable name, the molecular clamp.
Which sounds like something from a hardware store.
It really does. But the idea is actually pretty intuitive. A lot of COVID vaccine designs focused on the spike protein, the part of the coronavirus that sticks out and helps it latch onto our cells. If your immune system learns that shape, it can respond faster if the real virus shows up.
The catch is, when you take that spike protein out of the virus and try to work with it in a lab, it can be finicky. It doesn't always hold its shape. And if it changes shape, the immune system might learn the wrong picture, so to speak.
Right. So the molecular clamp was designed to hold the spike protein in the right configuration, basically to stabilize it so it stays in the form the immune system needs to recognize.
Like, if you're trying to take a photo of something, and it keeps moving, you'd want to keep it still. Same concept.
Now here's the part that becomes internet fuel. To make this clamp work, they used a tiny fragment of a protein that had been studied in HIV research.
And that sentence by itself, without the rest, is basically the whole problem.
Because people hear HIV and think infectious virus. But what they used was not HIV, not a complete virus, not something that can replicate, and not something that can cause infection. It was a small protein fragment used as a structural tool, like a piece of molecular scaffolding.
In other words, it's closer to saying, we used a tiny component that's been useful in HIV-related lab work, not, we included HIV in the vaccine. Those are wildly different claims.
So early lab work looked promising. The stabilized spike protein seemed to trigger an immune response. And that's when the project moved toward a phase 1 clinical trial in 2020.
Phase 1 is the early stage where the main questions are, is it safe, and does it do what we think it does in the body?
Exactly. Volunteers stepped up under strict oversight and monitoring. And then the researchers noticed something unexpected. Some participants were getting positive results on certain HIV screening tests.
Which, let's be real, would scare anyone if you got that call. Like, people sometimes hear false positive and think it's a small thing, but emotionally that's huge.
Totally. And from the research side, it's also a big red flag that needs immediate investigation. So they dug in. What they found is the key point. The participants did not have HIV. They were not infected. Instead, their immune systems had made antibodies that reacted to that clamp fragment. And some HIV tests, particularly screening tests, look for antibodies, not the virus itself.
So the test is basically saying, hey, I see antibodies that look like they could be related to HIV exposure, and it flags it. But it can't tell why those antibodies are there unless you do more specific testing.
Right. When researchers used more advanced diagnostics, tests that check for the actual virus rather than just antibodies. There was no HIV virus in anyone. The positive results were false positives caused by the immune response to the vaccine design.
I think this is where the internet story gets twisted. People leap from HIV test positive to HIV infection when those aren't the same thing. The test result is a clue. It's not the entire conclusion.
And here's where the project hit a real-world wall. Even though nobody was infected, the false positives were not a minor inconvenience. Imagine rolling out a vaccine to millions of people, and afterward a chunk of them triggers positive results on certain HIV screening tests.
That would be chaos for public health programs. Those quick screening tests are used all the time for early detection and treatment pathways. If the tests get noisy, you're creating confusion for clinics, stress for patients, and extra burdens on systems that already have limited resources.
So even though the vaccine candidate wasn't giving anyone HIV, it was interfering with a major diagnostic tool used worldwide. And that's not something you can just shrug off.
This is the part where people sometimes say, why cancel it if it worked? But worked isn't just made antibodies. It has to be workable in the real world, with all the ripple effects.
Exactly. After reviewing the data and considering the consequences, the team at the University of Queensland and the Australian government decided in late 2020 to discontinue that vaccine candidate. Funding was pulled, the project ended, and it never went into broad public use.
And I want to underline that this vaccine was not rolled out to the population. It didn't become part of the mass vaccination campaigns you saw later with other vaccines.
Meanwhile, globally, other approaches were moving fast. mRNA vaccines, viral vector vaccines, and other protein-based designs. Within about a year, several vaccines had cleared large trials with tens of thousands of volunteers and showed major reductions in severe illness and deaths.
So the molecular clamp effort became one chapter in a massive story. And to me, it's kind of an example of the system working. You test, you find a problem, you stop.
Yes, clinical trials aren't just a hoop to jump through. They're a filter designed to catch issues early. In this case, the issue wasn't that people were harmed by HIV infection. It was that the design could mess with HIV screening, which is a serious unintended effect.
So where does the viral claim come from? Like, why did this suddenly turn into COVID vaccine caused HIV?
This is where media context matters. In 2021, the BBC documentary series Horizon did an episode about the global race for COVID vaccines. It included Professor Chappell talking about the molecular clamp and how they used that tiny HIV-related protein fragment as part of the design.
And in the full documentary, it also explains that the project was abandoned, and it explains the false positive issue, right?
Exactly. In the complete segment, you get the whole arc. Clever idea, unexpected diagnostic complication, responsible decision to halt. But online, a short clip started circulating, basically just the HIV fragment part without the rest.
So if you only see the clip, you think you've uncovered something. They used HIV in a vaccine. And then people add captions and commentary, and suddenly it's framed like a cover-up.
Right. And this is a common pattern. Short video fragments move faster than nuanced explanations. The internet loves a shocking detail more than a careful timeline.
And once that narrative gets traction, corrections feel boring. Like, actually it was a protein fragment used as a stabilizer, and it caused false positives on some antibody-based screening tests, and there was no virus present is not exactly clickbait.
But that boring version is the accurate version. Fact-checkers, scientists, and journalists addressed it directly. The participants were not infected. The candidate wasn't distributed, and the positive screening tests were false positives.
Also, it's worth pointing out that false positives aren't unique to this story. Screening tests, by design, sometimes trade a bit of specificity for speed and sensitivity. Then you confirm with more definitive diagnostics.
Yes, and in this case, the confirmatory testing showed no HIV infection. The issue was the antibody reaction to the clamp component.
Another detail I've seen people miss, even the volunteers' false positive antibodies didn't necessarily last forever. Over time, those antibody levels can fade, and people can return to normal screening results. But the key point is still the same: no HIV virus was ever detected in them.
So what's the big takeaway? For me, it's two things. One, science is complicated and sometimes messy, and that's not a scandal. That's reality. 2. Communication is fragile. Remove context, and you can turn a cautious research decision into a scary conspiracy headline.
And it also shows why "this got cancelled" isn't always proof of wrongdoing. Sometimes it means the safety systems and practical considerations worked. A project can be technically interesting and still not be suitable for the real world.
Right? Thousands of medical ideas get tested. Most don't become products. That's not evidence of failure in the system. It's evidence the system is filtering, refining, and protecting people.
So if someone tells you, a COVID vaccine used HIV and gave people HIV, the accurate response is, there was an Australian vaccine candidate that used a small, non-infectious protein fragment as part of a stabilization method, and it led to false positives on some HIV antibody screening tests. No one got HIV from it, and the project was stopped before it could be widely used.
That's the story, start to finish. Not a secret infection. Not a hidden experiment unleashed on the public. A trial, an unexpected diagnostic complication, and a shutdown before broader rollout.
Which is honestly kind of reassuring if you let it be. It's scientists under pressure, trying ideas, checking results, and choosing caution when something doesn't fit.
And next time you see a clip that makes your stomach drop, ask, what's the missing minute before this? What's the missing minute after? Because context is usually where the truth lives. That's it for today's episode of Web Conspiracy. If you want more of these breakdowns, where we take a viral claim and rebuild the full timeline, send us what you're seeing online, and we'll dig in.
And please, if you only take one thing from this one, false positive screening test does not equal infection. It's a starting point for investigation, not the final verdict.
We'll catch you next time.